Hello everyone, I Assistant Professor Pragati Shete from Department of Microbiology from KG Somaya College. In the last online lecture, we started the topic of the biochips. In the last lecture, we saw the biochip, the definitions of the biochips. Then we saw the features, what are the unique features of the biochips that makes it the so special and the so convenient, efficient, right? Next we saw some of the major uses, the most famous uses of the biochips. Then we saw the biochip components. What are all the components present in the biochips, right? So in that component we saw the first one, that is the transponder. And transponder is again divided into the four parts. The four parts, namely computer microchip, right? We saw the functioning of the computer microchip, then the antenna coil, tuning capacitor, and the glass capsule. So these four components are present, four parts are present in the transponder. Okay, we saw each functioning of the each parts of the transponder components. Okay, now next we will see the reader or the detector, which is the second major component that made up the biochip. Okay, then reader, as the name says, it will read the signals. Okay, it will read the signal and it will convert that signals in such a way that it will display the result digitally on the screen. Okay, so now this reader is also consisting of the exciter coil, which produces an electromagnetic field. Okay, this coil will produce electromagnetic field via the radio signal. Now these radio signals, the previous slides we saw the capacitor and the reader are both tuned on the same frequency of these radio waves. So if you noticed, we can say that the radio waves are used in the processing or in the system, biochip systems. Okay, so now this capacitor and the reader are also uses the same frequency of the radio waves to establish the communication between the both. Okay, now coming to our reader part. So the reader will consist the exciter coil, and this coil will create the electromagnetic field via the radio signal. So using the radio signal, with the help of the radio signal, what will this exciter do? The exciter coil will produce the electromagnetic field, and which provide the necessary energy to activate the implanted biochip. Okay, this electromagnetic field will provide the energy, and this energy is necessary to activate the implanted biochip. The reader will also carry a receiving coil, right? So two coils are present in the reader, namely the first is the exciter coil and the second is the receiving coil. Okay, now the receiving coil receives the ID number. So I mentioned earlier, in the transponder's first part, that is the computer microchip, the computer microchip will have the 10 to 15 long digit identification number, right? So that identification number is received by the reader. In the receiving coil, okay, the reader will also carry a receiving coil that receive the ID number sent back from the activated implanted biochip. Okay, now the reader will also contain the software and components that will decode this identification number, or we can say the code. Okay, and display the result on the digital screen. Okay, so we can say the reader will contain the three parts: exciter coil, receiving coil, and the software. Okay, exciter coil, what will do? Exciter coil will produce the electromagnetic field with the help of radio signals. Okay, and this electromagnetic field, that is with the produced with the help of the radio signal, will provide the necessary energy to activate the implanted biochip. Okay, so the exciter coil will excites or will activate the implanted biochip. Next come the receiving coil. Once the biochip is activated, it will carry out its biochemical reaction, biological reaction. Then it will produce some signals, right? That signal will receive by the receiving coil. Okay, that receives the signals. Signals are in the form of identification numbers, right? As I mentioned earlier, the identification number on the computer microchip. Okay, that identification or the codes will be received by the receiving coil. Okay, now the third is the software. The reader will also contain the software and the components that will decode, that will carrying out the processing, the decoding of the received code, the received identification number, and display the signal on the digitally screen and display the result. Okay, you can see in this picture there is the digital screen, right? So the reader will have the software, that software and components that will decode that code, the identification number, and display the result. Okay, so next is the implanting of the biochips. So these are the two major components of the biochips that are made up of, biochips are made up of. Okay, first is the transponder, which is having 4 parts. Okay, 4 parts I mentioned earlier. And the second component is the reader. Okay, reader is also having the 3 parts. So this is about the construction, this is about the components of the biochips. Biochip is made up of. Okay, then next we will see the implanting the biochip, how we can insert this biochip, right? We saw the applications and the uses of the biochip. The main step is to how to implant this biochip, how to insert this biochip. So the biochip is inserted into the subject, right, into the subject with a hypodermic syringe. Okay, with a hypodermic syringe, and this injection is very safe, which means it will not cause any side effects on the skin or anything. Okay, comparable to the common vaccines. Okay, the anesthesia is also not required for the implanting this biochip, for injecting this biochip. Okay, so we can say that this is very easy to implant without causing any harm effect or side effects, right? So the hypodermic syringe we are using, the hypodermic means this will go immediately beneath the skin. Okay, that is called as the hypodermic. Dermic means skin. Okay, so inside the skin. In the last slide, in the previous slides, you have saw this picture, one picture of the hand in which the biochip is inserted, right? So that was example for the implanting the biochip. You can refer that also. Okay, so the biochip is inserted into the subject, into the person's body, or into the subject, any subject that we want to insert our biochip is, with a hypodermic syringe. The injection is very safe and simple as compared to the common vaccines. Okay, as we know, after we take the vaccine, we may have certain side effects, right? Certain rashes or the fever or the uneasiness. So like that, there is nothing in case of the biochip implantation. Okay, also along with this, anesthesia is even not required with the implant or for the implantation of the biochips. Okay, next is the working of the biochip. We saw the working in the part of the biochip components, also the construction of the component biochips also. Now we will see the grossly. Okay, the working of biochips starts with the reader. The reader generate a low power electromagnetic radiations via the radio signal. Okay, in the earlier slide we saw the reader having the exciter coil. Okay, that exciter coil is producing the electromagnetic field with the help of the radio signal. That electromagnetic field provide the energy to start to activate the implanted biochip, right? So the first step is what? The reader generate the electric field, okay, via the radio signal. And now this radio signals, this low power electromagnetic field is generated. After this, implanted biochips get activated, right? As I mentioned, the implanted biochip gets activates. Now the biochip will send the ID code. Okay, the first two steps are for the readers. The third step is for the transponder. The ID number is present in the computer microchip. The computer microchip is present in the transponder, right? So now the biochip, once the biochip is activated, the biochip will send the ID code, ID number back to the reader with the help of the radio signal. So if you noticed, we can say the processes or the signal transfers is carrying out via the radio signals, right? Every signal is receiving or sending via the radio signals. So now the biochip is activated, biochip is sending the ID code back to the reader via the radio signal. Again, now the role of the reader will come. Reader will amplify the received signals, right? In the reader, the second coil, that is the receiving coil, that will receive this code, receive the ID number. Okay, and now the biochips will the reader will also having this software. Now with the help of the software, it will convert the digital convert this code, ID number, ID code, to the digital format and displays on the LCD. Okay, on the digital screen on the LCD. So this is the working of the biochip. The biochip will work like that once inserted. Okay, so how our biochip is getting activated? With the help of the reader. Okay, reader generating the low power electromagnetic field with the help of the reader coil, okay, via the radio signals that will activate the biochips. Okay, biochip implant. Now once the biochips, the transponder will activate, that will send signals, that is ID number, back to the reader. Okay, and that signal ID number will be received by the reader with the help of the receiving coils. Okay, now these signals are received by the with the reader, right, by the reader. Now what will the bioreader do? The reader will have the software, right? That software will convert this ID number, this ID code, to the digital format, digital components, such that it will get displayed on the digital screen on the LCDs. Okay, so this is the working of the biochip. Next is the types of the biochips. Now we saw the components of the biochips, the construction of the biochips, and the working of the biochips, right? The working of the biochip. Now we will see the types of the biochips. So there are many type of the biochips. Most commonly, DNA chip also can be called as the lab on a chip, DNA chip, then the protein chip, enzyme chip. So the biological molecule in these chips, as for example in the protein chip, the protein is present. In the enzyme chip, the enzyme will carry out the reaction, right? So like that, there is the various types of the biochips. Okay, DNA chip, protein chip, enzyme chip, lab on a chip. Lab on a chip example is the DNA microarray. You can see in this picture the DNA microarray, which is a very important method, technique in the biotechnology, in the molecular biology. Okay, what is the DNA microarray? It is the two-dimensional array of the molecules. Okay, two-dimensional array on a solid substrate like the glass slide, or the, you can see in this picture there is the glass slide. Okay, the glass slide or the silicon film cell that will test the large amount of the biological materials substrate using the high throughput screening. Okay, and with the help of DNA microarray also we can carry out many reactions at the single time. Okay, it is a two-dimensional array placed on a solid substrate. DNA molecules are present, okay, placed on a microarray on the two-dimensional array placed on a solid substrate, mostly a glass slide or the silicon cell. Okay, that will carry out the large amount of the biological material using the high throughput screening methods. Okay, now in this picture on the microarray, DNA microarray, you can see there is the DNA microarray in the blue color cell. This is the healthy cell. Okay, and if we want to determine the presence of the infection or the disease causing cells, okay, then we can use this DNA microarray technique. Now blue color is the healthy cell and the purple color is the pathological cell. Okay, now if the cells are present, it will produce, if the pathological cells are present, it will produce the signals. That signals will be detected by the detector or the reader. Okay, in the DNA microarray, the fluorescence substances, the fluorescent materials are used such that if the pathological cell is present, that will emit the fluorescence, so that we can detect the presence of the pathological cell in a sample. Okay, you observe the dot diagram. In that is the DNA microarray. Okay, in that the black color or the gray color circles indicates the cells which are not present. Okay, not present in cells. Green color present in the normal cells. Okay, yellow color is present in both the cells, healthy cells and the pathological cell, and the red color is present in the pathological cell only. Okay, so what we do the signaling, we will add the fluorescent molecules that will be very specific to each cell. Okay, in this the red color fluorescent molecules will bind only to the pathological cell. Yellow color will bind to the both the cell, pathological also and the healthy also. Okay, and the green color cell will only bind to the normal cells. Okay, so now if the red color is present, we can detect the presence of the pathological cells. right? If the pathological cell present, it will, the biochip will sense the, detect its signals, right? And therefore the fluorescent molecule that is very specific for that pathological cell will bind to that, and that will create the red color fluorescence, that will detect, that will determine the presence of it, the pathological cell, in the sample. Okay, so you can see the red dots there, red, green and the yellow color dots that are present. And with the help of this we can also estimate, we can also count, right? We can count the cells, how many cells are present. So it is the very important type of the biochips. What it is? The DNA microarray. The DNA microarray, very important tool in the molecular biology to determine any genetic sequence, or to determine majorly in the infection, in the disease cells. Okay, so like this we have the DNA chip, that is the DNA microarray chip, protein chip, enzyme chip, implantable chip, right? In the previous slides we saw the implantable chip, the implantation of the biochip. Okay, so we will see the each type of the biochip in the next lecture. I hope you understood this lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.